Knockout organisms are basically silencing genes. Genetic modification does not always include adding a active gene to the genome of organism. It's not necessary that you have to add a gene. You can also remove a gene. That is why it is called as the knockout gene. In some situation, scientists want to remove a silent genes. Obviously some genes are mutated, कुछ genes इतनी mutated होती हैं कि they became no not responsive, हमें visibly उनका कोई काम नज़र नहीं आ रहा होता है. So we can call them as the knockout gene. They are used widely and are very important in genetic research. Now, in a knockout organism, scientists silence or basically knockout genes. They can make mute or silent one or more genes which are no longer functioning or we don't need them any longer. Okay, earlier we used to feel or we have studied that some human have had some sort of tail right on their back and we don't need them, na? So that could be, if that gene is present nowadays, that could be called as the knockout gene. They do this by inserting by the new gene similar to a gene which is to be investigated but makes the original gene impossible to read. So they are going to manipulate makeup of body in a way that body is not going to let that gene active. So that will be muted automatically or silenced. As a result, the original gene is silenced and cannot make a protein. Obviously it will go non-functioning. Knockout genes are usually accompanied by marker genes to show how they have been incorporated. So marker genes are basically kind of identification gene. We are going to impose them or attach them to the genes so that we could identify, like, okay, among these 20 to 25 genes, the middle one who is showing a bit light onto it is the knockout gene, and we have to silence it, and then ultimately, if not needed, we can remove it. Researchers can use knockout organism to identify the function of a gene genome sequence. Knocking them out and observing them, the result can help make their function clear. So first thing is that you need to make them silent. You need to put a marker on them. When you place a marker on them, it is definitely a sign that you don't need them any longer. The third case you are going to do is, we need to identify the function. Obviously when you have just put a marker onto that particular gene, you would have place any identification mark right on that gene, so it will make you feel like either this gene is performing any function or either you can directly cut it off. So gene sequence identifies many genes, but scientists can often do not know the function of gene in an organism. So knocking them out will make it clear, like either removal of that gene had any negative impact or positive impact, or either no impact. So we can also use knockout gene to investigate organisms disease. Obviously now this technology is on the way to us. It's not familiar. Somehow we can make a prediction, like, okay, if somebody might develop genes of cancer in him, we can knock it off, but it is going to take lots of time, lots of researches. But till now, so far, this has been limited to only the non-functional gene, because there is a massive percentage in the human genome which is non-functional. So in order to read the functional genome, scientists are preferring to cut off the unwanted or non-functional gene, and now they call it as the knockout genes or knockout organisms. So I'm gonna give a summary. Knockout gene is simply a gene which has no function in the human body or any living organism. We first need to silence it down. Upon silencing it, we can check out whether its function is affected or not. If no, it means we can remove it quite easily. So genome sequences identify the genes who are non-functional, then we can remove that gene. So this was it for the knocking out organism or knocking out genes. Subject vocabulary is basically helping us to figure out what the genetic engineering is, what is the genetic modification and genetic editing, it's all the same thing. So insertion of any
Okay.
gene from one organism into another organism, into by changing somehow its genetic makeup, because if we want to remove some disease from an individual, if we want to remove any infection, we can also go through it. If we want to add any development or helping or aiding genes particularly, we can add into it. Just like we have studied, if there are two cows, one is known for giving meat, other is known for giving milk, we can make gene editing, we can fuse them up through the DNA, recombinant DNA. We will make a recombined or recombinant DNA, that will be combination of the two, and that they will be going to help us to create a new individual with both the characteristics. Now, reverse transcriptase is the one. It's an enzyme which is used to make artificial copies of the desired gene by taking an mRNA molecule transcribed from the gene and using it to produce the correct DNA sequence. The reverse transcriptase is an enzyme which is used to prepare artificial copies of the desired DNA. Again, milk giving cow. Now we can take example of it. So you can take the gene from that desired organism. That messenger RNA will be responsible for making copies of it, and then that molecule will be responsible for transcribing the copies. Next thing would be sticky end. The sticky end, what is it? Basically, they are the two irregular pairs or strands of the DNA. The name given to the area is a base pair. One side of the base pair is left longer, other is left smaller, so that they could make the bonding with the other DNA available. So that is helpful to add the new piece of DNA. Replica plating is when a process used to identify a recombinant cell that involves growing identical patterns of the bacterial colonies on plates with the different media. So they are basically those plates, those stuffs who are helpful to develop the copies bacterial colonies. Obviously they would be needing diet, they would be needing stuff to maintain their progress basically, or they maintain their growth basically. Okay, gene gun is the one, it's a technique to produce a recombinant DNA by shooting the desired DNA into the cell at high speed, or on very small gold tungsten belts. Obviously we will be using those guns. By using those guns we can speedily insert those gene into the sample individual liposome wrapping, to avoid unwrapping of the DNA, unwrapping of the recombinant DNA. We will wrap DNA into these things, because obviously as soon as the DNA is going to reach different parts of the body, every single part is having bit variable pH. So it must be reaching that particular organ where it should have to reach. So we will obviously the DNA of that particular material will be aware, like either it have to open into the acidic media or the basic media. Micro injection is a technique used for producing recombinant DNA that involves injecting DNA into a cell through a very fine micropipette. We are well aware of it. Last one is the knocking out organism. These are those organisms in which one or more genes are got silenced and they are no longer working. So we have to first mute them up, we have to first silent them up, and then we can see either is their function to be performed by them. Most of the time they are performing no functions, so we can separate them. And that's how we are done with the chapter.